Okay, so this is the chemical formula of cellular respiration right here. Okay, so you get 6 molecules of glucose, so C6H12O6 plus your molecules, 6 molecules of oxygen to give you ATP, water, carbon dioxide, and some form of energy, or some energy in the form of heat that is released after any chemical reaction. We talked about this before spring break. Heat is released in some form or fashion, okay, even if it's a small amount. Now, glucose is your best friend. Glucose is the model molecule of cellular respiration, okay? So in order to get ATP or energy, we have to catabolize glucose, okay? So we have to actually go through a catabolic reaction of glucose in order to get that ATP. So, examples here. If you think about it, you eat this ice cream, okay? Ice cream cone, we get sugar in there, which is a carbohydrate, so that gives us glucose. From there, we have to add in oxygen that we breathe, okay? We breathe in oxygen, in case you didn't know. Now our enzymes come in, they help speed up this chemical reaction, so that way we can actually get some energy out of it. Okay, now in order to add an enzyme, yes, we have to use ATP. So some of the ATP that our body had already made it previously that was stored helps create and add the enzymes for a chemical reaction. Once we add in that oxygen with that glucose, we can then produce water, carbon dioxide, because that is what we breathe out, and ATP. Okay, so if you think about a combustion reaction, so like burning wood to make a fire, you're making a lot of heat energy. Okay, so you're burning fuels in one quick step, whereas respiration, you're burning it in many small steps. So combustion is in one quick step. Here's my wood, okay, I automatically add flame to it, okay, and boom, we have fire. I'm making a lot of heat energy. All I had to do was add fire. So how do we harvest energy from the fuels that we take in? So digesting your large molecules into smaller ones. That's just as simple as that. So you break the bonds from your more complex molecules, and that way when you do that, you're releasing the energy that was built to create those bonds, and you're moving the electrons from one molecule to another. So as the electrons move, they are negative charge, so they are carrying that negative energy with them. And that energy is then stored in another bond when they come in contact with another molecule to bond with, okay? So when that energy is stored in another bond, it can be released as heat if they are broken up, or it can be harvested to make our ATP later on in the process. Okay, so we kind of got a brief introduction to oxidation and reduction last time. So if you are losing or giving up the electron, you're being oxidized. If you are gaining the electron, you are being reduced. Remember, Leo says GER or oil rig. Okay, so oxidize is losing the electrons, and then reduction is gaining the electrons. So how do we move the electrons in biology? So electrons can't move alone in cells. They can't just decide, hey, I want to jump over here, so let's go ahead and move. So in order to do that, they have to move as part of a hydrogen atom. Because if you remember, hydrogen only has one proton and one electron. Okay, so they move in the form of a hydrogen atom because that is the equivalent of one electron. Okay, so it kind of uses it to move around. So if you look at our glucose and our oxygen, okay, so our glucose, since it's going from C6H12O6 to getting broken down now to carbon dioxide, we are losing a hydrogen electron, okay, from glucose and giving it, okay, to water. So we're losing that electron, that hydrogen atom, going to the oxygen that we combine with, okay? Since the oxygen is gaining that hydrogen, it is becoming reduced and it's becoming water. Glucose is being oxidized because it's losing the electron and becomes carbon dioxide, okay? So that's how we move through our cellular respiration. So if we're talking about our redox reactions in respiration itself, the whole focus is to break your carbon to carbon bonds. Okay, glucose has a bunch of carbon bonded together. So we're kind of breaking those down. So we're going to strip off the electrons from the carbon to hydrogen bonds by removing the hydrogen atoms. So glucose, we have 6 carbons and 12 hydrogens. So basically 2 hydrogens per carbon. We're stripping those carbon atoms from or the hydrogen atoms from the carbon to create our CO2. So that means that the fuel has now been oxidized. So our glucose, our fuel, our energy has been oxidized because our electrons have been removed. Now the electrons are going to be attracted to the more electronegative atoms. Oxygen is a very electronegative atom. So that is our most electronegative atom in biology. We've talked about that before. So that means that now our loose electrons from glucose are going to be attracted to oxygen. So now we're going to create water from that, and our oxygen has been reduced. So here's a quick breakdown review of oxidation versus reduction. Adding oxygen, removing hydrogen, loss of electrons releases energy, exergonic for oxidation, the complete opposite for reduction. This is just a review. Now, to move the electrons in respiration, the way that our electron carriers actually move the electrons is basically by shuttling the hydrogen atoms around. So we have these molecules called NAD plus and FAD plus 2. So NAD plus, here we are adding a hydrogen to it, which means it is gaining an electron, so it is being reduced. FAD plus 2 gains electrons for our hydrogen, so it's being reduced. Okay, so these are important molecules for the process of cellular respiration because they're going to be moving our electrons around, so that way we can make sure the correct oxidation and reduction is happening at the correct step of cellular respiration. So here we see we have ATP, right? Okay, so here our hydrogen is getting moved and it's coming over here. Our NAD plus takes that H and becomes NADH and is being reduced. So it's moving and carrying the electron itself. Okay, so that way it can be reduced. So think of NADH kind of as our electron piggy bank. Okay, that's all where it's going to go. All our electrons are going to go there to kind of carry it and move it around where it needs to go. So, overview in general of cellular respiration. This diagram right here is a great overview model of cellular respiration without all of the crazy hectic details that I am about to show you. So in your notes, or if you left your notebook at school, like on a piece of paper or something, I highly suggest you draw out this diagram here because this is the simplest way that you are going to see it from here on out. So we have multiple steps. We have 4 stages to go through cellular respiration. First step is glycolysis. Okay, we have glycolysis, which is anaerobic respiration. Anaerobic means without oxygen. Okay, so we don't need oxygen for glycolysis. Glycolysis occurs in the cytosol, okay, cytosol or cytoplasm of the cell. Then step 2, we're getting into aerobic respiration. So the last 2, 3, and 4 steps for cellular respiration are aerobic, which means they all occur with oxygen. The only one that does not occur with oxygen is glycolysis. So aerobic respiration, the rest of the steps occur inside of the mitochondria. We remember the mitochondria as the powerhouse of the cell, okay? Mitochondria, that's where we make all of our ATP from our glucose. So that's why cellular respiration occurs here. Step 2 is called pyruvate oxidation. Step 3 is the Krebs cycle. And step 4 is the electron transport chain. You could also see electron transport chain shortened as ETC. Okay, I'm going to break down each of these systems and processes for you guys coming up. So, stage 1, glycolysis. We already said that it's anaerobic, okay, but it can also happen aerobically. There's just very specific situations for that. So, glycolysis, you guessed it, has to do with glucose. Oh my gosh, okay. So we are breaking down glucose, glyco, sugar, lysis, splitting. So when we do this, our glucose is breaking down to become pyruvate. Now, this happening in the cytosol or the cytoplasm is very important evolutionarily, because in evolution, if you think of our very, very old original OG cells, what did they have? Did they have a lot of organelles, or was it basically kind of just, their DNA just floating around? They had their DNA just floating around. They didn't have complex organelles. It was just cytoplasm. So we kind of needed this process of breaking down our sugar to happen in the cytoplasm or in the cytosol, because we didn't have mitochondria to go through this. So this is the ancient pathway that energy was harvested. Okay, so this is where energy transfer first evolved. So they still had to take in organic molecules to create ATP. It's still the starting point for all cellular respiration. The only problem is it's very, very inefficient. You only get 2 ATP for every 1 molecule of glucose. Now, in your notes on the piece of paper, whatever, the amount of ATP you are using for each, or the amount of ATP that you are getting for each step of cellular respiration is very important. What you are putting into the equation and what you are taking out, those are the top things you need to know. So glycolysis, for every 1 molecule of glucose, you only get 2 ATP. That's a whole bunch of work for not a lot of payment. So this is basically describing what I just said with your prokaryotes having no organelles, okay? And then you also have to think that atmosphere did not have oxygen. Remember, we had our Miller and Urey experiment to create the original atmospheric gases, okay? There was not a lot of oxygen, so everything had to happen anaerobically. So prokaryotes that evolved glycolysis are ancestors of all modern life. They're the real OGs, okay? So glycolysis summary. We have our energy investment. Okay, glucose comes in, we get 2 ATP. So the energy investment we add in to glucose. That's we're investing in our creation of ATP. The payoff that we get is 4 ATP. But guess what? Out of that 4 ATP, we needed to use 2 ATP to actually break down the glucose. So that is why our net yield is only 2 ATP. Now, since we are getting rid of remember our glucose, we're breaking it down, we're getting rid of those electrons to form carbon dioxide. So because of that, we need our electron carrier, our little electron piggy bank to stash in some electrons. So we have NAD plus coming in to take in 2 of those electrons, so that way we can get our 2-pyruvate. So glucose becomes this molecule called pyruvate plus water. We get 2 ATP, 4 in total, but we had to use 2 to break down glucose, so we only end up with 2. And we get 2 molecules of NADH because they helped carry some of our electrons from our glucose breakdown. So this is the net yield and the important stuff. So it's endergonic, okay? We're getting energy into to eventually release it. Here's exergonic because we're harvesting a little ATP and a little NADH, okay? And then here we're going to kind of go over the substrate level phosphorylation with our kinases, and I'll do that in part 3.